All right, diving in today, getting into the nitty-gritty, the really crucial stage of bringing new meds to folks. We're talking phase 2 clinical trials.
Yeah, you can picture it like, think of it like this super important fact-finding mission, right?
Okay, yeah.
We've seen, some promising hints, some glimmers in the lab, some early data, and now we're like, all right, let's take the show on the road.
Mm-hmm.
Let's see how this drug actually works, how it behaves, but this time it's in actual people who are dealing with the condition we're aiming at.
Right.
It's like, are we on the right path here? Does it have the potential to help? But also, are there any surprises, any bumps in the road we didn't see coming?
Exactly. Got to find those out early.
Absolutely. Phase 2 is where the rubber meets the road.
It's that make or break moment.
It really is. We start to see, does this thing actually work like we hoped it would in the people we want to help?
Yeah.
But equally important, we got to be super thorough checking for any short-term side effects, any risks, and figure out the right dose to use if we're going to do bigger studies later.
Right. Got to get the dosage right.
You got it. And the information we get from phase 2, that's gold. Seriously. It's like the foundation for everything else. It basically decides if this drug keeps going, if we keep developing it, or if we have to head back to the drawing board.
High stakes. And for today's deep dive, you've really gone all out on the research. Transcripts, excerpts, books.
We went deep. We got into the science of how drugs actually interact with the body. We're talking pharmacokinetics here, all the way to the regulations, the rules set out by the big dogs, the FDA and the ICH.
The FDA, right, and the ICH, the International Council for Harmonization.
That's the one. And we even touched on how they design drugs at the molecular level, medicinal chemistry, and how digital tools are, shaking things up in the process.
Oh, we've got the full picture.
The whole enchilada. And our goal today is to walk you through the systems they use, how they keep tabs on everything during these trials, how they capture all that crucial data, and what they do when something unexpected pops up, those rapid response strategies.
Okay, I like that. Rapid response.
You got to be ready, because in phase 2, patient safety is the absolute top priority, no question. But at the same time, we're trying to find those early signs that the drug is actually doing what it's supposed to, that it's having a positive effect.
It's a balancing act for sure.
It really is. A delicate dance.
All right, so let's rewind for a sec. We've talked about preclinical testing before, all the lab work, animal studies, that sort of thing. But why is the monitoring in phase 2 so intense? Like, what actually changes when we go from the lab to testing in real people?
Great question. Because even though those preclinical studies give us a ton of valuable info about safety, is this thing toxic? Phase 2 is the first time we're putting this drug into a much more complex system.
Meaning the human body.
Bingo. And not just any human body, a human body that's dealing with the specific disease we're targeting.
Right, which adds another layer of complexity.
Exactly. And there are all these variations, these differences between people that we just can't fully replicate in the lab or in animals.
Like what? Give me some examples.
Sure, think about genetics, right? Everyone's got a unique genetic makeup. That can affect how their body processes the drug.
Makes sense.
Then there's just the overall way people's bodies work, their physiology. Some people might absorb the drug faster than others, some slower.
And people are taking other medications, have other conditions too, right?
Exactly. So all of that can influence how a drug gets into the body, how it spreads around, how it gets broken down, how it gets eliminated. We call this whole process pharmacokinetics.
Pharmacokinetics. It's like a whole journey the drug takes through the body.
It is a complex journey with many stops.
Okay, so each person's body is handling the drug a little differently. What does that mean for safety?
Well, it means there's always a chance that something unexpected could pop up, something we didn't see in the animals.
Even if the drug seems safe in the lab.
Even then, because sometimes there are differences between species. What's safe for a mouse might not be safe for a human, or a side effect might only show up in humans.
That's a bit unnerving.
It can be, but that's why phase 2 is so important. It's where we start to get a real understanding of the drug's safety profile in the actual people we want to treat. We're building that safety net.
Right, and you mentioned the FDA and the ICH before. I'm guessing they have something to say about all this.
Oh, absolutely. They've set up these really strict guidelines for safety monitoring during phase 2. It shows how important it is to get this right, to balance the need for data with the absolute priority of patient safety.
It's like a tightrope walk.
You got it. We're always on the lookout for those early signs that the drug is actually helping, that it's doing what it's designed to do, but we can never, ever compromise on safety.
Gotcha. So let's get specific here. What are the researchers actually looking at? What kind of data are they gathering during phase 2 to make sure everything's okay?
Okay, so there are a few key areas. First up, adverse events. We call them AOEs.
AEs, all right.
Basically, anything that happens that's unfavorable, unintended, like a bad sign, a new symptom, a health condition,
Sure.
anything like that that happens during the trial.
So it doesn't even matter if they think it's caused by the drug.
Doesn't matter. We write everything down. It's like
Okay.
if you get a headache during the trial, that goes in the report.
Even if it's just a normal headache, not because of the drug.
Even then, might seem like overkill, but here's the thing.
Okay.
Later on, we do all these fancy statistical analyses. And let's say, just for example, we see that more people taking the drug get headaches than people taking the placebo. Right.
Okay, so a pattern emerges.
Right. It could be a total coincidence, or it could be an early sign that the drug is having some effect we didn't expect, maybe on a different part of the body.
Interesting. So you catch it early before it becomes a bigger problem.
Exactly. That's why we got to be so thorough. And then, of course, we're checking those basic vital signs all the time.
All vital signs, yeah. Heart rate, blood pressure, all that good stuff.
All of it. Temperature, respiratory rate. We track it all at very specific times, according to the study plan. And that lets us see even small changes that might be linked to the drug.
Right. Consistency is key.
Consistency is king. And we're not just eyeballing it, you know?
Right.
We're taking precise measurements using standardized procedures.
Makes sense. And what about blood work?
Oh, blood work is crucial. We do a bunch of lab tests. Hematology, that's all about your blood cells, making sure they're healthy, the counts are good.
Okay.
Then there's clinical chemistry, which tells us about how your organs are functioning, liver, kidneys, all the important stuff.
Right. Essential stuff.
Absolutely. Plus, we check electrolyte levels and other markers that can tell us how the body is reacting to the drug.
So is there a standard set of blood tests? Or does it depend on the drug and the condition?
Good question. It definitely depends. It's all tailored to the specific drug and the disease we're studying.
Custom-made lab panel, basically.
Exactly. It's not one size fits all. And beyond the blood work, we also do regular physical exams to get a sense of the participants' overall health.
Good old-fashioned checkup.
Can't beat it. And something that's become increasingly important is what we call patient-reported outcomes.
Patient-reported outcomes. That sounds interesting. What's that about?
So this is info that comes directly from the patients themselves.
Okay.
We ask them about their symptoms, how they're feeling, how they think the treatment is affecting their daily life, that sort of thing.
So it's not just about the numbers, it's about how they're actually experiencing things.
Exactly. It's a really valuable way to understand the impact of the drug from the patient's perspective.
I can see that. What kind of things do you ask them about?
All sorts of things. We might ask them to rate their pain levels, tell us about their fatigue, or use questionnaires to assess their quality of life, their ability to do everyday activities.
Right, how it's affecting their life overall.
Exactly. And then, of course, we also have what we call the efficacy endpoints.
Efficacy, meaning, is it actually working?
You got it. These are the specific measurements that tell us if the drug is having the effect we want it to have on the disease.
Okay, so if it's a cancer drug, the efficacy endpoint might be tumor shrinkage.
Exactly. Or if it's for Alzheimer's, it might be improvement on certain cognitive tests. It really depends on the disease and how we measure improvement.
So it's all about picking the right measurements, the right endpoints, to see if the drug is actually making a difference.
It's all about the endpoints. And the key is choosing ones that are sensitive enough to detect subtle changes, especially in early-stage trials. Sometimes those changes might be small, but they can still be meaningful.
That makes sense. So it sounds like you're gathering a ton of info from all these different sources.
A ton. It's a whole holistic approach, looking at the big picture, not just one piece of the puzzle. We want to see how the drug is affecting the whole person, not just their disease.
Right, and you said before that everything is tailored to the specific drug and the condition?
Always. There's no cookie-cutter approach here.
Gotcha. So let's talk logistics for a second. All this data you're collecting.
Yeah.
How do you actually capture it and manage it all during a phase 2 trial? It seems like it would be a nightmare to keep track of everything.
It's a lot, that's for sure. In the old days, it was all done on paper. Imagine these big, thick forms called case report forms, or CRFs.
CRFs, okay.
And every patient got one. Every time they came in for a visit, the researchers would fill out these forms, record all the data, every little detail.
So a whole stack of paperwork for each person.
Exactly. And then they had to store all those forms, analyze them. It was a huge headache.
I bet. And prone to errors, too, I imagine.
Oh, yeah. Handwriting mistakes, missing data. It was a recipe for disaster. But thankfully, things have moved on since then.
So what do you use now?
Now it's all electronic. We use these systems called electronic data capture systems, or EDC systems for short.
EDCs. So it's like a digital version of those paper forms.
Exactly. Now, instead of scribbling things down, the researchers enter the data directly into a secure computer system.
Sounds much more efficient.
It is. So much faster, less room for errors,
Right.
and way easier to analyze the data later on.
And I'm guessing there are rules about using electronic systems for medical data, right?
Oh, absolutely. There are strict regulations, especially in the US with the FDA.
Makes sense. So what kind of rules are we talking about?
Well, the big one is something called 21 CFR Part 11. It's all about making sure that electronic records are trustworthy, that they're accurate, and that they can't be tampered with.
So it's about data integrity, basically.
Exactly. It's like having a digital chain of custody for the data. So, for example, if a doctor enters a patient's blood pressure into the system, 21 CFR Part 11 makes sure that there's a record of who entered it, when they entered it, and if anyone made any changes to it later on.
So everything is tracked and documented.
Everything. And that's really important for the FDA. They need to know that the data they're getting is reliable and hasn't been manipulated in any way.
Makes sense. So these EDC systems are basically a game changer for managing data in clinical trials.
They really are. They've made things so much more efficient and accurate.
Okay, so we've talked about safety monitoring, but let's switch gears now and talk about the other big goal of phase 2, figuring out if the drug is actually working.
Right. We want to see those early signs of efficacy, those hints that the drug is having the desired effect.
So how do you actually track that?
Well, it all comes back to those efficacy endpoints we talked about before, those specific measurements that tell us if the drug is working.
Right, like tumor shrinkage or improved cognitive function.
Exactly. So throughout the trial, we're carefully monitoring those endpoints in the group of people getting the drug and comparing them to the control group.
The control group, right, the people who aren't getting the drug.
Exactly. They might get a placebo or they might get a standard treatment that's already available. And what we're looking for is a statistically significant difference between the two groups.
Meaning a difference that's big enough that it's probably not just due to chance.
Exactly. And not only statistically significant, but also clinically meaningful.
Meaning it actually makes a real difference in people's lives.
Exactly. So we're looking for changes in those endpoints that suggest the drug is having a real positive impact on the disease. And in some phase 2 trials, we might do something called an interim analysis.
An interim analysis, what's that?
It's basically a sneak peek at the data before the trial is finished.
Oh, interesting. So why would you want to look at the data before the trial is over?
There are a few reasons. First of all, if the drug is showing a really clear benefit early on, we might stop the trial early for ethical reasons.
Ethical reasons.
Yeah, because it wouldn't be fair to keep the people in the control group from getting a drug that we already know is working well.
Ah, I see. So everyone gets a chance to benefit.
Exactly. And on the flip side, if the data shows that the drug isn't working, or if there are serious safety concerns, we might also stop the trial early to prevent people from being exposed to unnecessary risks.
So it's a way to make sure that the trial is still ethical and safe even while it's ongoing.
Exactly. And those interim analyses can also give us valuable information that helps us adjust the trial design.
Adjust the design. How so?
Well, for example, we might decide to change the dose of the drug based on how people are responding.
Or maybe change who's eligible to participate in the trial, right?
Exactly. So it allows for a more flexible approach, making sure we're getting the best possible data from the trial.
Makes sense. It's like course correction mid-flight.
Exactly. And speaking of course correction, what happens if something does go wrong during the trial?
Right, like a safety issue pops up or it becomes clear that the drug just isn't working.
Yeah, those are the moments when you really need to have a plan. And thankfully, we do. We have rapid response strategies in place to deal with those kinds of situations.
Okay, so let's talk about those. What are the key elements of a rapid response strategy?
Well, first of all, it's all about being proactive. You can't just wait for something bad to happen. You have to be actively looking for any red flags.
So constant monitoring, basically.
Constant vigilance.
Yeah.
And then you need to have a clear plan for what to do if something does go wrong.
Like a step-by-step guide.
Exactly. Who needs to be notified? What information needs to be gathered? What decisions need to be made? It all needs to be spelled out in advance.
So there's no confusion or delay when something happens.
Exactly. You got to be ready to act fast. And one of the most important parts of the rapid response system is the Data Safety Monitoring Board, or DSMB.
DSMB.
These are like the independent watchdogs of the trial.
Watchdogs.
Yeah, they're a group of experts, usually doctors, statisticians, ethicists, who aren't directly involved in running the trial.
So they're impartial, objective.
Totally. And their job is to keep a close eye on the safety data as it comes in. They meet regularly, review everything, and if they see anything concerning, they have the power to recommend changes to the trial.
Changes like?
They could recommend lowering the dose, changing the eligibility criteria, even stopping the trial altogether if the risks outweigh the benefits.
Wow, so they have a lot of authority.
They do. They're there to protect the participants, first and foremost. And alongside the DSMB, there are also ethics committees that oversee the trial. They make sure everything is being done ethically and that the participants' rights are being protected.
So multiple layers of oversight to ensure safety.
Multiple layers. You can never be too careful when you're dealing with people's health. And if something serious does happen, like a participant has a severe reaction to the drug, there's a clear procedure for reporting it to the authorities.
The authorities meaning?
The FDA in the U.S. or other regulatory agencies around the world.
Right, so they can investigate and take action if necessary.
Exactly. And sometimes, based on what we learned during the trial, we might need to make changes to the trial plan itself, the protocol.
So the protocol isn't set in stone, it can be adapted.
Absolutely. We might need to adjust the dose, change the inclusion criteria, add new safety monitoring procedures. It all depends on what the data tells us.
So it's a dynamic process, constantly evolving.
It is. We're always learning and adjusting based on what we're seeing. And those protocol amendments are crucial for making sure the trial stays safe and scientifically sound.
Okay, this has been really helpful, breaking down all the different aspects of monitoring in Phase 2. Now, our research for this deep dive covers a pretty broad range of topics, right?
But well wide, yeah.
And they don't always give specific examples related to Phase 2 monitoring. But do you think we can connect some of these broader ideas, some of the concepts we've explored, back to this crucial stage of drug development?
Oh, for sure. We can definitely draw some parallels. Like, remember we talked about how drugs are metabolized, broken down by the body?
Yeah, and those CYP enzymes, right?
Yeah.
Like CYP3A4.
Exactly. So let's say in preclinical studies, we find out that our drug is mainly broken down by a specific enzyme. Well, in Phase 2, we're going to be really careful about monitoring drug levels in patients' blood.
Why is that so important?
Because people can have different levels of those enzymes, thanks to their genes or other meds they're taking. And those variations can make a big difference in how much drug is actually active in their body.
So someone might need a higher dose, or maybe they need a lower dose to avoid side effects?
Exactly. So understanding those pharmacokinetic processes, how the drug is absorbed, distributed, metabolized, eliminated, that's really key for interpreting the Phase 2 data, both the good stuff and the bad stuff.
Right. It helps you make sense of why people are responding the way they are.
Exactly. And we also talked about those structural alerts in medicinal chemistry, remember?
Vaguely. Remind me.
Okay, so basically, there are certain chemical features in a drug molecule that can make it more likely to form toxic byproducts.
Oh, right. Those red flags in the drug structure.
Exactly. Now, ideally, we'd identify those red flags early on, even before Phase 2.
Right.
But Phase 2 is still a time to be extra vigilant, especially if the drug has one of those structural alerts.
So what kind of extra monitoring would you do in that case?
Well, we might do specific lab tests to look for signs of organ damage that could be caused by those toxic byproducts. You're basically playing detective, looking for any clues that something might be off.
Interesting. And we also discussed how medicinal chemists try to tweak the drug's structure, maybe by changing those heterocyclic groups to make it more effective.
Yeah, that's a big part of what they do.
But could those structural changes also affect safety?
Absolutely. It's all connected. They're not just trying to make the drug more potent, they're also trying to make it safer.
So they're aiming for that sweet spot, maximum effectiveness, minimum risk.
Exactly. And those tweaks to the drug structure can definitely influence what we need to watch out for in Phase 2.
Okay, so the design of the drug itself is feeding into the safety monitoring plan. And of course, we can't forget about the FDA and the ICH, those regulatory bodies you mentioned earlier.
The regulatory landscape, always a key player.
Yeah, and their guidelines seem to really shape how Phase 2 trials are run. Can you tell us a bit more about that?
Sure. So the FDA, before they even let you start testing a drug in humans, they want to see a ton of data from those preclinical studies,
Mm.
the pharmacology, the toxicology.
Right, they want to make sure it's not going to kill anyone.
Exactly. And that sets the stage for really rigorous safety monitoring once you do start testing in people.
So those preclinical studies are like the foundation for everything that comes after.
Absolutely. And then you have all those ICH guidelines, like good clinical practice or GCP. That's like the Bible for how to run clinical trials ethically and scientifically.
Okay, the rule book, basically.
Exactly. And there are specific guidelines for safety pharmacology, too, that outline what kinds of safety tests need to be done early on in drug development.
And those tests would inform what you're specifically monitoring in phase 2, right?
Exactly. They help you pinpoint what to watch out for. And then there's ICHQ9, which is all about managing risks.
Risk management?
Yeah, it emphasizes being proactive, thinking ahead about what could go wrong, and having plans in place to prevent it or mitigate it. And that applies to everything in drug development, including phase 2 monitoring.
So it's not just about reacting to problems, it's about anticipating them and trying to avoid them in the first place.
Exactly. And all these regulations, all these guidelines, they're there for a reason. They're there to protect the participants and make sure that the data we're getting from these trials is reliable and trustworthy.
So it's a highly regulated environment for good reason. It's all about balancing that pursuit of new treatments with the absolute priority of patient safety.
It's a delicate balance, but it's absolutely essential.
Okay, I think we've covered a lot of ground here. For our listeners who might be new to all this, let's try to summarize the key takeaways about monitoring safety and efficacy in phase 2 clinical trials.
Okay, so the big picture is this. Phase 2 monitoring is all about walking that tightrope between carefully evaluating the safety of the drug in people who have the condition we're targeting and getting those first clues about whether the drug actually
Yeah,
works.
those early signals of efficacy.
Exactly. And that involves collecting a ton of data, adverse events, vital signs, lab results, patient-reported outcomes, all of it. And we use sophisticated systems to manage all that data, make sure it's accurate, and analyze it properly.
And it's not just about collecting data passively, right? There's this constant vigilance, this active search for any signs of trouble.
Always on the lookout. And we have those independent watchdogs, the DSMBs and ethics committees, to keep an eye on things and make recommendations if needed. And if something does go wrong, we have those rapid response plans ready to go.
Inform the authorities, adapt the trial protocol, whatever it takes to protect the participant.
Exactly. And all of this happens within a very specific regulatory framework, those rules set out by the FDA and the ICH.
So it's a carefully controlled and constantly evolving process.
Very much so. And phase 2 is a crucial filter in drug development. It helps us figure out which drugs have the potential to be safe and effective and which ones we need to abandon.
Right, it helps us make those tough decisions about which drugs to move forward with and which ones to leave behind.
It's a critical stage. It really determines the fate of a drug. And it raises a really interesting question, as we move into the future of medicine.
Okay, what's the question?
Well, we're seeing all these new, really complex treatments being developed, biologics, gene therapies, drugs designed by AI. And it makes you wonder, how are we going to adapt our monitoring systems, our safety protocols to keep up with these advances? How do we make sure we're still protecting patients while also fostering innovation?
That is a great question. It's like the next frontier of drug development.
It is. It's a challenge, but it's also an exciting time.
Well, I think we've given our listeners a lot to think about today. Thanks for walking us through this complex and crucial world of phase 2 clinical trials.
My pleasure. Always happy to dive deep.
And for our listeners, thanks for joining us on this deep dive. We'll be back next time with another fascinating topic from the world of science and medicine.